Okay, so here we go. We are going to do a psychology sociology practice passage, and here is my method for getting through it, right? First off, for these passages, unlike cars, where you want to like read it thoroughly and like get an idea of the main idea, in this case, you want to just skim it and get a general idea. The reason is because most of the passage, like when you're doing the questions, that's going to tell you where you need to focus on. So skimming the passage is more time efficient for this section. But then you spend the time that you saved by skimming thoroughly understanding the charts, especially the axes and the data points of the chart. Like you really want to have a good understanding of the charts because they are definitely asking you a question about it. And then once you feel like you've got a good idea, you start going through the questions and you use process of elimination as we worked through before. So let's go ahead and start here. Can you guys still see the browser right now or no?
Yes.
Yes, okay. Okay, good. So I remember last time I said that, last time I told you guys to just read it, and then a lot of people said they were kind of falling asleep. So I think this time I will just read it out. Do you guys want me to read it out or you guys want to try reading it yourself?
Quick question before we start reading. Do you advise that we read the question and go straight to the chart, or actually read the question, read the passage before trying to answer?
You should absolutely read the passage first in in almost every situation, because the thing is, reading the passage can only provide you additional context, right? Like, oh, actually, that's a very good question. Let me table that question for a second, okay? Let's or actually, why don't we try it? Let's try that method, right? Of going directly to the chart, right? Let's see what happens. So in this thing, let's say we quickly skim it and we're just like, significant dietary and nutritional differences can be found among racial and socioeconomic groups in the United States. These nutritional behavior differences are the foci of many studies, since they contribute to racial disparities in the incidence and prevalence of chronic disease and premature morbidity. Okay, so we already have like a decent amount of information. Dietary and nutritional differences across racial and socioeconomic groups. Okay, let's say that we are rushing and let's try to go directly to the chat, through the chart. Okay, so if we look at the first question, according to table 1, which of the following is true? On average, among Blacks, those in the low SES group had less healthy dietary behavior compared to those in the medium SES group across all time periods. So this chart is showing us socioeconomic status, right? So we see low, medium, and high, right? So we understand that they've segmented the socioeconomic status into 3 groups. And on the left side, we see that this chunk is representing white people, and this chunk is representing Black people, right? And here we have a bunch of smaller things, right? So first of all, we need to understand those in low SES group had the less healthy dietary behavior. That's the parameter in question, the less healthy dietary behavior. So which of these things on the left could represent less healthy dietary behavior? Your options are the number of respondents, the mean DQI score, the score of 4 or less, or a score of 10 or less. So out of those 4 options, what do you guys think is the relevant choice here?
I think DQI stands for dietary something. That's probably in the passage, but I think mean DQI score.
Okay, so let's say it's mean DQI, right? And let's look around, let's see if we can find a general idea of what mean DQI is. So here we see DQI, diet quality index. Okay, that seems good, right? So it seems like we're on the right track. Now, let's go back to evaluating the answer choice. On average among Blacks, those in the low SES group had less healthy dietary behavior compared to those in the medium SES group across all time periods. So what are the main 2 things that we're comparing? The low SES group and the medium SES group, right? And they're saying across all time periods. So when they say across all time periods, what do they mean? what here is representing time periods? We
Could
see
it be
rate.
those Roman numerals? Sorry.
Yeah, could it be the Roman numerals, right? That's a good guess because we don't really see anything else here about time. So we scroll up and we see, oh, 1965 is 1, 1977 is 2, et cetera, et cetera. So to observe trends over time. Okay, so we're putting the context clues together. So to compare, to compare across time, right? Let's compare the low SES group in, say, the first time period to the medium SES group in the second time period. And we're comparing on average among Blacks. So we go to this section and we see, let's see, in time period 1, the mean DQI score is 6.6. And in time period 2, the mean DQI score is 7.2, right? So what do we see here? Those in low SES groups had a less healthy dietary behavior compared to those in the medium SES groups. So is this true? This in the low SES, it's 6.6. In the medium SES, it's 7.2. Does that match what the answer choice is saying? The low SES had less healthy dietary behavior. Does that make sense, or should I let me write it out in chat, right? In chat, let's write out the question is, or maybe let me write it by annotating. So the question is, we see that low SES has a DQI of 6.6 in time period 1, right? And medium SES has a DQI of 7.2 in time period 1, right? So does the low SES group have less healthy dietary behavior than the medium SES group? So please contemplate this question for a minute, and then I'd like to see your answer in chat. I'm trying to find a good, or maybe I'll change the color. Not sure where to put this. Are we assuming that a lower DQI score means less healthy? That's the question. So did you answer yes to the question itself?
Yeah, yes to the question and to the question Fatal asked.
Okay.
Because I think it says diet quality index, and sort of intuitively, the quality of something correlates to like less quality, less numbers, more quality, higher numbers.
Right. So what does everybody think? Does the low SES group have less healthy dietary behavior than the medium SES group? And we only compared 1 point, right? You can look at the other points as well across the time. So we compared 7.2 to 6.6, then you compare 6.7 to 6.5, and compare 6.4 to 6.2. And we see consistently that the medium group has a higher value than the lower group. So what I would love to get some responses. Do you guys think that the lower SES group has a less healthy dietary behavior than the medium SES group?
Yes.
All right. Is there anybody out there for no? Is there anyone who's not sure? Okay, so the question that Fatu asked is very important. Are we assuming that a lower DQI score means less healthy? And this is, I'm glad that Damilare asked about this approach because it seems very intuitive that DQI should mean that the higher it is, the better quality, and the lower it is, the worse quality, right? Because that's something that intuitively makes sense. But you have to understand that this test is designed in such a way that your intuition will work against you because they don't want you to intuitively move about things. They want you to critically think about things. And intuition relies on shortcuts and, mental heuristics, right? They want you to algorithmically go through things and really break it down and know that you had a good understanding of it. So the chart first approach doesn't work because the chart is not entirely encompassing all of the information you need. This is very, very common because we need to go back to we need to go back up ahead and we need to read about the DQI. What does a DQI mean? The primary outcome was a score 0 to 16 on the Diet Quality Index, a composite of 8 food and nutrient-based recommendations from the National Academy of Sciences, including areas such as eating 5 or more, blah, 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 right? Look at how much useless filler information is here because the intent is to get you to get your eyes to glaze over and for you to stop paying attention. But right here at the end, a total score of 4 or less was considered to indicate a more healthy diet, and a value of 10 or more indicates a relatively less healthy diet. Right there at the end, they flipped everything on you. All of your intuition is now out the window. You now need to flip it, right? So you asked a very important question. Are we assuming that a lower DQI score means less healthy? The MCAT is not really going to ask you to assume anything. You're not really going to be assuming it. I mean, you'd be assuming if you're trying to guess something that you can't remember, that you need to remember, but you're not really supposed to assume anything. So overall, does that make sense why reading the questions and then jumping to the chart or something is not a good idea because you can miss things in context that you need. You can miss things in context even if you read, but you have less of a chance if that happens. So I'm glad that we did it that way to see how we can get tripped up. Yeah, it was a good example because it's just thrown in there right at the end of the second paragraph, right? Amongst a bunch of baloney that you don't need to know. So but unfortunately it's incredibly important. And my recommendation is whenever you see something like this, you need to really take note of it and write it down because even if you notice it, this is something that happened to me a lot. Even if you notice it and you're like, oh, okay, I get it, right? The problem is the intuition is so powerful that, quality equals more equals better, that it overrides your critical thinking. Like in the back of your mind when you're reading, you're like, oh, okay, I'll remember that. And then you forget it because it's like, it's like that the sky is blue and you read in the passage like, by the way, in this example, the sky is green, right? And then you probably just forget it when you're trying to rack with the questions. So if you see something like this, you immediately write it down. So I would have written down, if I was going through the thing, I would have written down somewhere okay, increased DQI means less healthy. That's it. That's as simple as you need to make it. Very simple, right? You can even go a step further and you can do I don't know how to draw a down arrow. Maybe I'll just make one like this. Yeah, there we go. And then I can write the opposite. Decreased DQI equals more healthy. Okay, so that takes out a lot of mental guesswork for you. You can just write that and then just refer to that, and then that will help you. Okay, so now let's go back and let's do this by reading or skimming first, and then approaching the questions. Is everybody with me so far? Everybody following? Any questions that you want to ask? We can just use the thumbs up reaction. You don't have to type in chat or anything. Okay, seems like everybody's good to go. Let us continue on. Okay, right. So let's read through this. So we had read the first part already. That was just an introduction to the ideas of the passage. Using survey data of over 60,000 participants collected by the U.S. Department of Agriculture, Study 1 compares dietary trends among 32,406 Black and white non-pregnant adults 18 years or older of varying socioeconomic status. So if I was doing this, I would probably highlight socioeconomic status, and then we would see, because that's just like a keyword. How do I know it's a keyword? Because when you're studying, you're going to read a lot about socioeconomic status and stuff like that. The primary outcome was the score 0 to 16 on the diet quality index. I'm not going to read this, superfluous information again. For each recommendation, a person could score 0 to 2. A total score of 4 or less was considered to be indicative of a more healthy diet, and a value of 10 or more indicates a relatively less healthy diet. Again, whenever you see them introducing a brand new concept and explaining it, write it down in a way that makes sense to you, in a way that's very easy to digest. So you don't need to waste time going back to the passage and being like, wait, what did it mean again? And rereading it. Don't just write it down once and then you have it. So categories of SES were based on education and income. Respondents with over 12 years of education and an income level over 350% of poverty level were categorized as high SES. Those with less than high school education and income less than 185% were classified as low SES. 3 time periods were taken into account, blah, blah, blah. Observe trends over time. Table 1 displays some results. We're not even going to look at the table now. I'm going to finish reading, and then I'm going to go back and deeply dissect the table. More recently, study 2 adjusts for SES and directly explores the association between race and nutrition. So notice here, study 2. So we've moved past. This is a different study now. Using data from blah, 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 a researcher examines the difference between the nutritional behavior of Blacks and whites. N equals 3,350. If you don't know, N means the number of the subjects, right? The researcher attempts to observe whether Blacks differ significantly from whites in terms of health-related nutritional behaviors that have established associations to the development of heart disease, cancer, stroke, and diabetes. The results indicate that even within the same SES group, whites are more likely, on average, to exhibit healthy nutritional behaviors such as the consumption of at least 5 fruits, et cetera, et cetera, et cetera, right? We see all this stuff, but it's whatever, we get it. Exhibit healthy nutritional behaviors. That's what I mean by skimming. You don't need to go into every single, because they're just going to throw out as much detail as they can because they want to trip you up, right? But the main thing is to get the relationships, right? Whites are more likely on average. This is less likely. This is less healthy. This is more healthy. Those are the important relationship defining things that you need to dig out of the passage, and not waste your time reading about fruits and dairy products and high fiber. That's not the important part. Okay. For categories such as consuming whole grain products and consuming being, no significant differences were found. Okay, whatever. And then we have sources, right? The sources, I have never seen this be useful. I'm sure it could be in some way. It could be, but I've never really seen it be useful, to be honest. So it's I'm not sure what scenario it'd even be useful to read this. I'm not sure. The jury's out on that one. But now we've skimmed this. Let's get back to trying to answer these questions, right? So according to table 1, which of the following is true? And so we dissected the first one pretty thoroughly. And we see that the thing is that now that we know this relationship, right? They're saying those in the low SES group had less healthy dietary behavior. So the low SES group among Black individuals. So 6.6 to 7.2, right? We see that a lower DQI means more healthy. And when they're saying less healthy, boom, this is wrong. Okay? Does everybody understand why A is incorrect? Or if anyone doesn't understand why A is incorrect, then just say why. Say you need explanation. Okay, seems good. Option B, comparing Blacks and whites in low and medium SES groups, the proportion. Oh, so the other thing is when we we could spend all day here reading through this chart very carefully and being like, okay, so the difference between the low and medium here is this and this and this, and between the low and high is this. But that's a waste of time because you only need to evaluate as much as the question asks you. Okay, so you're not writing a lab report, you're not critiquing an article or anything, right? You just need to get the information, extract it, and go, right? So it's enough to have a general idea of what this chart is telling you, right? And the idea is, okay, we're comparing Black individuals and white individuals on the basis of their diet quality index. That's enough. You have a good understanding of what this chart is telling you now. Sorry, I have to include and across socioeconomic status. Now I have a good understanding, a complete holistic understanding of the chart. And the specific data comparisons will do when prompted by the questions. So comparing Blacks and whites in low and medium SES groups. So we go, first we position ourselves on the chart. Blacks and whites, so bottom and top, in low and medium. So we're only dealing with this area, right? The proportion of Blacks with poor diet quality exceeded that of whites in each time period. The proportion of Blacks. If we're talking about the proportion of Blacks, what from these labels on the left, where are we looking? Which row should we be looking at? The proportion of Blacks.
Number of
Go ahead.
respondents.
Yeah, the percent, I think it's percent of respondents. Yes. So that is the proportion of Blacks with, so we're looking at the proportion of Blacks with poor diet quality, and we're seeing, does it exceed that of the white proportion, right? So from comparing in low and medium, right? So in low, their proportion. So here's a question, right? Which of these? Is it score 4 or less, or is it score 10 or more? Which of those correlates to poor diet quality? If you have a DQI of 4 or if you have a DQI of 10, which one corresponds to poor health? And here, instead of going back and digging through the thing, we have written down a rule for us, right? Score 10 or more, yes, because we wrote down a rule, increased DQI equals less healthy. So we're going to be looking at the score 10 or more section and comparing it in both, right? So comparing Blacks and whites in low and medium SES groups, the proportion of Blacks with poor diet quality exceeded that. So here we have 6.1%. So right, this is not a DQI, right? This is a percent of responders. What is the DQI for this 6.1? This 6.1%, what is their DQI?
$6.6.
Where do you see 6.6? Oh, this one, right? Okay, but okay, this 6.6 represents the mean DQI score, the average, right? This 6.1% is those who scored 10 or more. And this 16.4 is those who scored 4 or less. So that's why we're only looking at we're only comparing this row to this row, because these are the only people who with poor diet quality. That's what they're asking for. So it's very careful it's very important to carefully dissect the chart, I guess. So this 6.1% to this 10.7%, right? Let's start there. The proportion of Blacks with poor diet quality exceeded that of whites in each time period. Is that true? Is 6.1% greater than 10.7%? No. So we can already say that this is false, right? We could keep going. We see 5.0, 6.7. Again, it's wrong. Does everybody get why that's wrong? Because they're asking the proportion of Blacks with poor diet quality exceeded that of whites in each time period. But that's not true. The proportion was lower compared to white people. Okay. Dietary quality improved over time for both blacks and whites across all socioeconomic status. So here we will be comparing over time, right? So where do we look at to compare over time? This column to this column to this column, right? Because this is going from 1 to 2 to 3. And we saw earlier that 1 to 2 to 3 represents the time periods from 1965 to 77 to the 80s, right? So we need to see, so what pattern do we need to see if we need to see dietary quality improving? If the dietary quality improves, what happens to the DQI?
Numbers go down, get smaller.
Correct. The numbers go down, right? So if you're thinking that way already, then you'll be able to identify the pattern easier, right? If you answer it conceptually for yourself already, then you'll be like, okay, let me try to find if the numbers go down, right? 7.2, 6.8, 6.3. Numbers went down. 7.1, 7.7, 7.1, 6.6. Numbers went down. 7.8, 7.3, 6.4. Numbers went down. So far, in the white column, it makes sense. 6.6, 6.5, 6.2. Again, numbers went down. 7.2, 6.7, 6.4. Numbers went down. Blank, 7.3, 6.0. Numbers went down. Because all the numbers managed to go down, we can say that actually this is true. The dietary quality improved over time. Notice how this is asking dietary quality improved over time, but you could easily replace this with the DQI decreased over time. And both of those sentences mean the exact same thing. This is what the MCAT does 100% of the time. They want you to understand the variable that they're working with, but the variable represents something. So that's always what they do. They ask about the representation rather than the variable itself, because it's easy to look at it and say, okay, the DQI is going down. So, okay, I would obviously pick the answer choice that says, oh, the DQI is going down. But they want you to take it a step further. Rather than asking about the DQI itself, they're asking about what the DQI actually represents. Okay, so from there, we can confidently say that C is correct because it's nothing wrong with it. But of course, we will evaluate D just to be safe. Differences in the mean DQI score according to race and SES have widened over time. So how would we evaluate this? Take a moment, look at that, and think about how could we evaluate this? Meaning, what numbers would we look at the chart at and to verify this claim?
Can you ask a question again?
Sure. So the question is, differences in the mean DQI score according to race and SES have widened over time. What numbers in the chart do we have to look at to evaluate the veracity of this claim? Like, how are we going to be able to tell if this is true or false? What numbers will tell us this?
So we're looking at the mean DQI scores.
Mm-hmm.
And
That's a
we're
good place to start. Yeah.
And we're comparing, let's say, low I in whites versus low I in blacks. And then we can go to like high I or actually not I. We can still stay in low and just go to a different time period, like triple I.
Yeah,
But the gaps have not changed that much, I think.
exactly. So what do you mean by the gap?
Like 7.2 to 6.6,
Mm-hmm.
and then 6.3 to 6.2 is like a few point differences. In fact, actually, it came closer. It didn't widen, it narrowed.
Boom, you got it. That's the main thing, right? We're observing a trend over time, right? And there is a trend. It's narrowing. Because of one word, this choice is wrong, right? Because they're saying the opposite. They're saying it's widening. So perfect. You looked at the choices, you identified the correct parameter, I guess, to evaluate, right? And then you looked at, what's actually happening over time. What is the relationship here? And you saw, oh, it's not widening, it's narrowing, right? You notice that there was a change. Describe the change, it's narrowing. That's not right. So that leads us all to C. So notice how, oh my God, we already spent 40 minutes on this. So
Crazy.
yeah, so notice how how much time you can spend. The reason we spend so much time on this is because this is what you have to do for almost every one of these passages. It's really about critical data analysis. And I'm trying to give you guys the tools to be able to look at this by yourself and do it confidently with practice, right? Following a specific method and following basically what I'm saying is like this takes a lot of time the first time because it's like the difference between giving a man a fish versus teaching them to fish, right? I'm trying to teach you guys to fish so you guys can catch your own fish. Okay, dumb metaphor aside, this is yeah, C. So again, we see just like we did, they wrote the relationship. The lower the DQI score, the better the diet, right? And for each SES group, the average DQI score. Okay, so that's fine. Does anybody have any questions about not just this question, but about tackling the chart, tackling the data, thinking critically about the data? Or any questions so far? Okay, I'm gonna move on then. Consider the results from both study 1 and study 2, assuming statistical significance for all findings presented, which of the following conclusion is best supported? In study 1, results indicate that on average, poor whites have better dietary behavior than rich whites. Again, this is basic comparison, right? So we go poor whites, right? We have the poor whites group, the low SES, versus the rich whites, right? And if the poor whites would have better dietary behaviors, they would have a lower or would they have a higher DQI? Well, according to our rule, they should have a lower DQI, right? So let's look. Do they have a lower DQI? 7.2, 6.8, 6.3, 7.8, 6, 7, 7.8, 7.3, 6.4. They do have a lower DQI, right? Let me see. Study 1 results indicate that on average, poor whites have better dietary behavior than rich whites. Poor whites, whoop. Poor whites
I think they have a less healthier if the numbers went up, like from 7.2 to 7.8 and a 6.8 to 7.3.
Well, they're saying poor better than rich. So these guys having higher numbers means they're less healthy. So the poor guys are right?
Gotcha.
I think so. Yeah, I think so. So it seems like A is correct for now, but we keep going. In study 2, the researcher finds that poor whites have better diets than rich whites. So again, study 2, don't get confused, right? Keep track of, another thing. If you see like study 2, study 1, highlight it so you can keep track. Because whenever they ask about multiple studies, they're always going to ask you to compare them. So you just highlight that and bring yourself back to study 2. Study 2, the researcher finds that poor whites have better diets than rich whites. I have no idea if that's true. I have to read this again. So what he found was the results indicate that even within the same SES groups, whites are more likely on average to exhibit healthy nutritional behaviors. So here's the thing, even within the same SES group, but he doesn't compare across SES groups, this study doesn't talk about poor and rich whites. So this is a thing that's important because if you miss study 2, you would probably end up comparing it here. But remember, this is study 1. This is not study 2. You do not cross-contaminate the studies is the best way for me to say it. So study B is incorrect because they only asked about study 2 and that's out of scope for study 2. C, in study 2, the researcher shows that blacks are more likely to have heart disease compared to whites due to the lack of positive dietary behaviors. They had said something about heart disease. They said, the researcher attempts to observe whether blacks differ significantly from whites in terms of health-related behaviors that have established associations to the development of heart disease, etc. Right? So he attempts to observe, And then what? Whites are more likely on average to exhibit healthy nutritional behaviors. And then that's it. This study doesn't make a comment about positive dietary behavior, doesn't make a comment about the heart disease. It does make a comment about the positive dietary behaviors. That's true. But it doesn't make a comment about heart disease, right? Notice that. Like they've thrown that in for you to intuitively be like, oh, that makes sense, right? That, if white people have better diets, then Black people are probably more at risk for heart disease. Okay, sure, that sounds good. No, you have to stay within the scope of whatever they give you on the passage, right? If they didn't explicitly mention it, then it's not something that you can again, they're asking which is best supported, right? There's no support for this in the passage. In either study, no health related racial disparities were found. Well, I mean, this is obviously just like wrong because, like they said right here, whites are more likely on average to exhibit healthy nutritional behavior. So they did find health related racial disparities, right? Racial disparities meaning differences across racial cohorts. So I think we found that A seemed to be the best answer. Let me see. Yes, okay. So it was A, and yeah. Any question? I know I kind of sped through that one because I realized we spent a lot of time on the first one. Does anyone have any questions about this one though? Am I going too fast or is it making sense?
No, this is a good pace for
Is it a
me.
good pace? Makes sense? Okay, good. Okay, so let's keep going. All right. If another researcher finds that strong social support among whites explains positive health behavior, which of the following is correct? Okay, so this is a very classic question about experimental literacy, I guess. Like again, so this is what we talked about before in the last class when we extensively went over that experimental design with the soap, with the germs in the soap, right? So here, what do you think is the independent variable and what's the dependent variable according to the sentence here? I'm hoping that everybody gets this right after you spent a lot of time last session. And this is not like a trick question. It really is that simple.
I have my mic unmuted, but I don't want to jump in and just say the answer.
I
Um,
mean, go for it. Unless anybody else wants to go, but I doubt it. You can go for it.
I'm thinking social support is the independent variable,
Hmm.
and then health behavior is the dependent variable. So D.
Yeah, it's as simple as that. A lot of this you may overthink it and be like, oh, I don't like, ooh, I might be screwing up. No, it's especially for behavioral science passages, the independent and dependent variable, like the experimental design of it is very straightforward usually. Yeah, it's that simple in this case. Socioeconomic gradients in health can best be explained by which of the following basic concept. So this is the other type of major question that you get in this section, right? Like I showed you guys last time, where you compare concepts that you have studied, like vocabulary, keywords, concepts that you've studied. So here, but the good thing is that even if you haven't studied them, or you can't remember them, a lot of them are very self-explanatory, right? So like the name itself gives you a decent idea of like what the concept means. So for this case, even if you don't know all of these, like what do you think it would be the best answer to this question? Socioeconomic gradients in health can be best explained by which of the following basic concept? Social stratification, intersectionality, class consciousness, or social exclusion? Anyone want to throw out a guess?
I'm thinking A.
Thinking A, social stratification? Yep, that's right. So social stratification is talking about, gradients and how things get distributed unevenly across social barriers or social divisions, right? Intersectionality is okay, here's another really, 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 really high value thing to do when you're studying, especially for this section, right? Every time you get, even if you get this right, do you know the rest of these? That's really important. Even if you got this right and you were like, oh, I'm happy, I got it right. You're not done yet. If you're practicing, you're not done yet. You need to check if you know what these things are, because you never know if it was a different question and the correct answer was actually one of these, right? That could be the case on test day. So it's not enough that you got it right when you're practicing. To truly be like, okay, I mastered this, you need to go back and make sure that you know the definition behind these other options, right? So what is intersectionality? Intersectionality is how, no man is just like one quality. Like, I'm not just Asian. I'm also a man. I'm also male. I'm also, somebody who grew up in a low SES neighborhood. I'm also someone who's bilingual. All of these things like mesh together to form a unique set of challenges and also privileges in my life, right? Somebody who has the exact same background as me, but they are a woman instead, faces much different, you know, scenarios and past history and stuff like that. So that's basically what intersectionality is getting across. Class consciousness is a term from Marx and from socialism. It's the idea that everybody is going to, like, the lower classes will wake up and realize that they're being exploited and that they have nothing to lose but their chains, and they will revolt against, the bourgeoisie. Social exclusion is just like, when you're excluded from, social exclusion happens to stuff like criminals and like delinquents and people who get pushed out of society, and can be linked to um, theories of deviance. Why do people act out? Why do people commit crimes and stuff? Can be linked because maybe they were socially excluded. So notice how I was able to deeply explain each of these other terms, right? Because you never know when one of these other terms would be the correct answer. That's really what you want to get into the habit of doing, being able to deeply explain each answer so you can feel confident that if you see it again, you're good to go with it. And yeah, okay. So any questions so far? This is like half of the questions on this section are going to be like this, at least half of them, because they just want to test your knowledge of the concepts. Good to go? Okay. Okay, so the last one. To further understand how SES influences health behavior and outcomes, we must think about the processes in which health outcomes occur. Which of the following is not a causal mechanism through which SES may affect health outcomes? So this is a very common type of question where it's not really actually related to the passage, other than the fact that this is about like health and SES and health, right? But it's not asking you to evaluate the data or any of the study. This is just asking about, it is just generally asking about something that's like sort of related to the topic. So you wouldn't even need to read the passage to answer this, but reading the passage would of course give you more context. So let's think. So obviously not a causal mechanism. To understand this, you would need to know what a causal mechanism is. A causal mechanism would be, what does it sound like? I don't know. I'm not sure for sure. I don't know what it means, but I know what causality is. I know that causality means that A causes B, right? It directly leads to B. So I'm looking for something that is not a direct A, if A, then B relationship, right? A person with higher SES is less likely to be exposed to bad environmental conditions that contribute to disease. Is this a causal mechanism? less likely to be exposed to bad environmental conditions that contribute to disease. So what are we seeing? We're seeing a person with higher SES is less likely to be exposed to bad environmental conditions that contribute to disease. So I I would say that this is a causal mechanism because we understand what is causing the disease. What's causing the disease? Bad environmental conditions. What's causing bad environmental conditions? Oh, sorry, less likely to be exposed to bad environmental conditions. Why are they less likely to be exposed? Because they have higher SES. Notice the link of the chain of causality, right? The flow of causality. We're understanding that this higher SES means you're less likely to be exposed to bad environment, means that you're less likely to get disease. A clear logic chain, right? A person with higher SES has more resources to buffer health threats. Higher SES leads to more resources, leads to buffering health threats, meaning buffering health threats means that you can, you know how like a chemical buffer, like if you have a chemical buffer, if you add acid or if you add base, it's not going to shoot up the pH or shoot down the pH. It will just be a gradual change because the buffer prevents a massive change. So if you have resources, you can prevent massive health threats. You can buffer against them. C, a person with lower SES is more likely to have mental health issues. This is, I think if you're able to see I think you can hear the difference between these. These guys, the first two explain what it is about SES that leads to the outcome, right? But this doesn't explain anything. This says lower SES, more likely to have mental health issues. But why? What causes the mental health issues? Had this said a person with lower SES is more likely to be exposed to violence that leads to mental health issues. That's causality, right? That's giving you the A, then B, then C. This is jumping from A to C. Does everybody see why this is jumping from A to C? I'm not talking about the answer choice. I'm talking about like jumping from X to Z instead of going X, Y, Z in terms of reasons. Okay. Does anybody not understand causality? Or like logic? Because this is a big thing. This is also a big thing for cars, especially. Um, okay. So it should be C. I mean, I'll look at the last one. A person with lower SES is less likely to have social. This is a good one. Like you can tell already, like, they're explaining thoroughly how they got to this. So yeah. But yeah, X to Y. Okay. So, all right. Let's take a quick 2-minute break. Oh my God, I don't even know if I'll finish the slides today. Let's take a quick 2-minute break and come back at 6 p.m. and start the biology slides. Yeah, okay. See you at 6. Okay, let's get right back into it. So for the biology section, it's it's a little weird, like what I can talk about, because a lot of the biology section is based on your own knowledge. And but there is a good amount of stuff that we can talk. So but for the first part, what I'm going to do is show you guys a lot of common things that you should focus on, I guess, and some mnemonics and tricks for you to remember that. Um, and then we will do a passage. I don't know. I don't know if we'll have time to do it today, but we'll see. But the biology section is very much like the content and the critical thinking is very hand in hand. You got to know the content so you can do the critical thinking. And it probably requires the most studying, I think. It's because there's just a ton of deep knowledge that you can acquire. So the first place we start is the amino acids. Does everybody have the amino acids memorized, or like are they working on it? I'm not sure like how often in undergrad you have to memorize. For me, my biochem professor was like, memorize your amino acids, you'll thank me later. But I don't know if everybody had to go through that. Um, but you need to
Yeah,
memorize,
definitely.
yeah, you need to memorize your amino acids. It's a huge, 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 huge part of the MCAT. It's probably the most like useful thing you can study, I think. I struggle to think of something that's like more useful because there's just so much, they can ask so many questions about it. But this is just one of my index cards from my Anki deck. And basically it includes everything that you need to know about amino acids, or almost everything. So you need to know which ones are the acidic ones, and you need to know which ones are the basic ones. They ask that all the time. That comes up all the time, especially in like the context of enzymes linking with substrates, and like they switch to basic one to an acidic one. What's going to happen? That kind of thing is very common. You need to memorize the structure. Yes, you need to memorize what they look like. You need to be able to draw them. Names, the one letters, the three-letter abbreviations, the properties. By properties, I mean like acidic, basic, nonpolar, polar, etc., and the pH. There's a lot to remember here, but it all is important and it's all um, actually useful. So the and here at the bottom is another little good tidbit to know. All naturally occurring amino acids are L, except glycine, which is achiral. Why is glycine achiral? Does anyone know?
It has two hydrogen. The side chain is a hydrogen instead of uh
Instead
like
of a group, yeah. Good. Yes. So if a carbon doesn't have four different substituents, then it doesn't have chirality, right? It's achiral. Of the 19L amino acids, all are S configuration except cysteine, which is R. So there are certain things like this where yes, you could like sit down and draw out the Kahn-Ingold prelog like system and like try to figure out the configuration if asked, but just memorize it and like save yourself the trouble. Like there are some cases where understanding how to do something is much less valuable than just memorizing it. Unfortunately, that's the situation, right? Like if you just like I can do it, but why would you want to do it on the test? You wouldn't. Just memorize it and like keep it in your head in case you need it, right? And there's going to be a lot of things like that where like getting to the answer is much harder than actually just remembering the answer. So that's something. Okay, and speaking about memorization, right? Here's my Google Doc that I use for memorizing, right? So I combine things by properties, right? So I wouldn't get them confused. So for nonpolar, I said Glenn, Alan, Valerie like international professional men and women's football. That made sense to me. I made like a little story out of it and I did it, right? The second one is about Pokemon. This probably makes absolutely no sense to most of you, right? And that's a good thing. Mnemonics are better when they are deeply ingrained to your thought patterns and thought processes. When they only make sense to you, it's because you created it. Like the better it fits with you and your personality and what you like and what you think about, the more it's going to be memorable for you. That's really the key of a good mnemonic. It makes sense? Okay, good. I I came up with this back in college, and I tried to share it with like my study buddies, and they were like, they looked at me like I had three heads, and I was like, okay, never mind. You figure it out then. Okay. The acidic one and basic ones I really like. The acidic one especially is incredibly good. Asphyxiation due to eating glue. Why is this so good? Because the D and the E are the one-letter abbreviations, and these are very unintuitive one-letter abbreviations. Because aspartic acid, instead of having A, because alanine has A, aspartic acid has D, and glutamic acid has E, because glycine has G. So this one is really, really helpful. I really like it. And it's acidic, and you know it's acidic because it's a horribly negative fate. So little things like little memory tricks like that are very, very useful. Especially in a high stress situation where you're like, oh shit, was this negative? Was it I lost it. Little memory tricks like that will carry you a lot. And the last one for basic ones is I couldn't think of it. Argus. I couldn't think of a better name. Argus reports his lying kids. Again, we match it. We match the one letter thing to like a weird one that doesn't really, like L is K, and that doesn't seem to make sense, but we match it in a way that makes sense to you. Again, if this doesn't make sense to you, make your own. In fact, it's better if you make your own. I'm just giving you this example as like a way for you to connect things. But ideally you should even make all of your own mnemonics, and that they will stay with you for life. But we don't always have time for that, so if you want to steal a mnemonic, feel free. This is another thing that you can just quickly memorize instead of like figuring out all the PI stuff, which is super annoying. This is about isoelectric point, and you can memorize that the PI is the average of the pKa's of the two functional groups, and you would use this rule. If the side chain is not basic, you use the lower two pKa's. If the side chain is basic, you use the upper two pKa's. Instead of like messing around with like, oh, what's going to be negative, what's going to be positive, and this try to make your life easy. Try to do rules that, like, whenever you can figure out a rule, be like, can I make something here that always works? And if it always works, memorize it and stick with it. I will upload these slides later, so don't worry about like, oh, I need to grab. Everything's going to be uploaded, and I will provide you guys with the slides. And also, I will provide you guys with the Anki deck that has all these cards. I just haven't gotten around to it. It's been a busy week. Any questions about amino acids? Probably not. I'm pretty sure everybody's like studying hard on amino acids anyway. But this is just to emphasize that you should study your amino acids real good, because they are very, very powerful. Everybody good?
Good.
Good. Okay. Also, yeah, you need pKa's for the occasional isoelectric problem. Okay, so now we'll move on to just talking about flashcards again. You see here, this is so we talked a little bit about this last time, but again, whenever I make a flashcard, I compare everything, because that's what they want on the test. They want you to compare, right? This microfilament, like just jumping a bunch of information about microfilaments on its own card, is not as useful as directly comparing it to the immediate neighbors, right? So if I have a card like this, I'm forcing myself to say, okay, microfilaments are composed of what? And then microtubulins are composed of what? And then intermediate filaments are composed of what? So it might seem like, oh, you're going to get it cross-contaminated, you're going to get it messed up. No, it's important for you to get it cross-contaminated now so that it won't happen on the test. What I mean is that if you get it confused, then you'll know, oh, I have this confused. I need to keep practicing it until I have it straight. And then when you have it straight, it'll be straight on the test, right? So yeah, the comparison. So my entire Anki deck that I used for this was made. I sorry, I wrote it in a way to compare everything always. And so you see that the parts that are occluded here, you see the parts that are in italics on the other options. So those italics would be occluded on like the next card. So the next card would compare the number 2s, and then the next card would compare the number 3s. The next card would compare the number 4s. And the reason for that is Obviously, if you look at this card and everything is hidden, you don't have the brain capacity to pull all that out. It's really hard. Instead, you can break it down into small facts. But if you ever need to just like look it up, everything is neatly organized on this, nice little card that has everything compared. So right here, does anybody remember, microfilaments, microtubules, intermediate filaments? Here's a real question. If you study this, can you answer this? Right? That's the real point. Don't be shy. If you get it wrong, it's just a learning opportunity.
Wait, what was the question?
Sorry, the question was like, can you fill in the blanks basically, right? So microfilaments are composed of what? Cytoskeleton.
Actin.
Microfilaments are composed of actin. Found near?
I don't know, the centromere or centrosome? I don't know.
I think so. I don't, but okay. So now I don't know if I've cross-contaminated because I haven't studied it in a long time. So we know that microfilaments, actin. So what is microtubules composed of?
Tubulin.
Tubulin. Yeah, that one's easy. And then where are microtubules found?
I know one of them is the kinetochore. So like when it pulls it
Mhm.
together and apart in anaphase. I mean, it's found in different places.
It is found in different places. I don't remember, so we'll see when we see it. Intermediate filaments composed of what? What are intermediate filaments made of?
Ah,
I think what are they made of? Oh, I remember. Okay, yeah, I remember. Yeah, this is just so we this you usually see the first two compared, but I threw this in there in case they ever compare it. And we have the cytoskeleton is a network of, blank that helps with blank and blank. So that's just
so
there.
I know it helps with shape and structure, but I
Mhm.
don't know what that first blank is for.
It's a network of a type of thing, right? A network of things. So let's take so this is the power of flashcards, right? You really have to
Yeah.
force yourself to know, like, hey, do I know? So let's take a look, right?
Yeah.
Network of protein fibers, right? If we were asked, what is the cytoskeleton? At the end of the day, it's protein fibers, right? And these are all different types of protein fibers, right? That helps with cellular movement and maintaining the cell structure and shape. So we got structure and shape. We may have forgotten about cellular movement. We may have forgotten about flagella, about cilia, right?
Ah, I see.
So
I see.
yeah, that's where you get the real like brain activation. You're like, oh, I forgot that. I won't forget that next time. That's really helpful. So microfilaments, you're right, composed of actin, found near the plasma membrane. I forget where I read this, but I read this somewhere. I need to review this again. I forgot why this is. I think this is because like it's for amoeboid movement. Yeah. Because it helps push the cell around basically. For cytokinesis, yes. Alpha tubulin, beta tubulin found near centrosomes, right? And then intermediate filaments composed of proteins like keratin. So we're talking about stuff like your nails and stuff because they do structural support. So I hope you guys can see like the value in structuring things like this, really forcing yourself to compare and contrast things before you have to do it on the test.
Yeah, this is a really cool method. Thanks for sharing.
Yeah, no problem. And I will share the flashcards themselves in when I get around to it. Oh man. Okay, so next, mnemonic, right? Enzyme kinetics. It shows up pretty decently. It shows up pretty often. So again, if you memorize things, it's helpful because you can always fall back on it, right? Yes, understanding is good. I'm not saying understanding is bad. You need to understand, but you also need to memorize in case you don't have the time to spare understanding. Okay. So you see this table here on the left. I really, really like tables for memorizing across like broad things, meaning that if you have a lot of things to compare, tables are really useful. Actually, like even if you think about it, this is like a loose table form because like if you could rearrange it to be like, what are they composed of? What do they form? What are they responsible for? This is like a table in a way. And the reason I like table is because humans actually have really, really good spatial memory, even more so than like, semantic memory or any other kind of just straight informational memory. Like we have very good spatial awareness. So when I make a table like this, I see the patterns and I memorize the patterns. So I know on this left one, it'll go up, it won't change, it'll go down, then it'll go up, it'll go down. Boom, that's a pattern I can memorize. Even if I forgot like why uncompetitive means we go down in CAM, at least I remembered, oh, up, then skip, then down, then up, then down again. Okay, I can remember that. Then I can remember for the second column, okay, we don't do anything at first, and then we just always go down. That's easy to remember, right? So I found tables to be really, really helpful, especially like, if I see the concept on the test, I'll be like, okay, give me 2 seconds for me to just write out this table. Well, not 2 seconds, give me 10 seconds for me to write out this table. And then I don't have to do like the mental legwork of like, what was uncompetitive again? How does mixed work again? None of that. Skip all that jazz. Just write it out. Just have like a mental schematic or table that you can just dump, right? Okay. So let me give you some mnemonics, right? So first of all, does everybody recognize what I mean by enzyme kinetics? Like this stuff, the how enzymes function thermodynamically. Is it thermodynamic? No, kinetically. Obviously it's kinetically, it's enzyme kinetics. Yeah, does everybody recognize this?
I do.
Okay. Okay, good. Okay. So the idea is let's talk about how we can easily remember this, how we can easily memorize this. So we have, instead of competitive, think competitive as in KM, right? That tells us if something is a competitive inhibitor, then the KM goes up. Okay, so that's our first memory hook. The competitive is competitive. So the KM goes up. If the KM goes up, so then what do you think would happen for a non-competitive? Well, no change on the KM because it's non-competitive, right? Then if it's uncompetitive, it would go down, right? Because we have 3 different options, right? Either it goes up, it doesn't do anything, or it goes down. So we know competitive goes up. We know non-competitive doesn't do anything, no change. Uncompetitive goes down. Okay, we've got it, right? Then we have mixed prefers enzyme versus mixed prefers enzyme substrate complex, right? The competitive, the KM is the substrate concentration at 1/2 Vmax, right? So here's where we're using our understanding to like understand our mnemonic a little bit better. The substrate concentration at 1/2 Vmax is the KM, right? If the KM increases, does that mean that if the KM is increasing, is the substrate able to bind better or is it binding worse? If that makes sense.
You said KM is the concentration of the substrate at 1/2 Vmax.
At 1/2 Vmax, yeah. So if the KM increases, so if the substrate concentration at 1/2 Vmax increases, is it binding better or is it binding worse? Like, is it easier for the substrate to bind or is it harder for the substrate to bind?
Vmax is not changing, right? It's just taking a
Vmax,
longer time to bind.
yes, yeah. The question is like, is it easy? Is it? I guess the stricter way to ask it is like, how fast are we reaching Vmax?
Well, that means it's if a KM increase, that means it's taking longer to reach Vmax.
Yes, exactly. So if KM increases and it takes longer to reach Vmax, does that mean that the substrate is the affinity high or is it low?
So the affinity would be low, right?
Boom.
If it's
Yes.
taking longer.
Yes, exactly. So we see here mixed inhibitor, but prefers the enzyme. If it prefers the enzyme and binds to the enzyme, then Km increases because it's now taking longer for the substrate to bind. Is that making sense? If it prefers the free enzyme, then the inhibitor binds the enzyme. If the inhibitor binds the free enzyme, the substrate has a harder time binding. Therefore, the affinity went lower, the Km went higher, and in this scenario, the Vmax decreased, just because this is the type of inhibitor. So logically thinking through that way can help you remember, like, oh, if this mixed inhibitor is preferring the free enzyme, then it must be blocking the substrate from getting on it. If it's blocking the substrate, well, what does substrate and Km relate? How do they relate? Well, I know if the affinity increases, then the Km decreases. And if the Km increases, the affinity decreases. If you cannot understand it like conceptually very well, or if you can't easily bring it up, then memorize it, right? Because you're not going to be able to have 100% understanding of every concept, right? Like there are some things that you'll like, especially me in like physics, right? Like there's a lot of things where I'm like, I'm trying really hard to understand this, but I don't understand it, right? And at some point I'm like, okay, I'm going to just memorize the relevant information that's enough to like get me through like the questions. Because like I have conceded to physics. Like I'm like, this is not the subject for me, and that's okay. So if you don't fully get it, as long as you can memorize the relevant information, especially with like a subject as broad as this, right? Because there's so many different concepts. If you can memorize the relevant subjects and put it down in a way that you can easily recall it, that's going to be really helpful for you. So this is how I memorize the effect of the inhibitors on the Km values and the Vmax values. Does anybody have any questions about this? Oh, actually, wait. Let me cover the other mnemonic. So if you get these Lineweaver-Burke plots, the way that you can remember it, competitive inhibition looks like two swords crossing each other. So it's like a competition. You see the blue sword and the red sword like fighting each other. Then uncompetitive inhibition is uncompetitive because you can draw a U between the lines. Let me see if I can draw it. Look right here. There's a U. Another thing to remember for uncompetitive inhibition is that the decrease in the Km and the decrease in the Vmax is united. It's an equal decrease. So the ratio will remain the same. Meaning that if before the Km, let's say the Km was 5 and the Vmax was 10. Then the change when it decreases, it will be like 2/4, because before it was 5/10, it was 1/2. So now it'll still be 1/2. That's a unique thing about uncompetitive inhibitors and how they affect the Km and the Vmax. So the change is united. Again, another memory hook. Then the last one, non-competitive inhibitors. The way I remember this is that they're not competing. They're not crossing each other, unlike competitive inhibitors, which are fighting each other, right? Does that make sense? A little, visual mnemonic.
Yep, tracking.
Okay, great. Let me get rid of this U. Okay, so I think that's the end of this part. Okay, so metabolism. So metabolism is the big boy of the biochem section. It's really the main content of the biochem section. I guess enzyme kinetics and biochem are the main stuff of biochemistry, and you kind of need to know it inside and out. Maybe not inside and out. Like you could get away with knowing just the major steps, and by major steps I mean major steps are any steps that involve control of the pathway. So what do I mean by that? Control of the pathway means that that's the step that can be influenced by other cofactors or activators or inactivators that will shift the pathway a different way. So let's say for glycolysis, right? We have the if we have a buildup of pyruvate, right, then we won't be continue doing glycolysis because because glycolysis produces pyruvate, and then pyruvate is like we will have feedback inhibition, right? Where there's too much of the product, so we don't need to make more of the product. But we also have at like fructose 1,6 bisphosphatase. I think that's the full name, right? The third one. At the third one, wait, let me see if I have a label. Yeah. At the third one, this guy has a lot of different regulators, right? Because once we do this and we break it down, right, we're kind of committed to this pathway. So before we fully break it down, we're like, hold on, do we really need to break it down? And the body has a lot of different activators and inactivators to say, okay, maybe let's do it, yes, or no, we shouldn't do it, right? Knowing those specific enzymes and knowing their roles is the most important part of metabolism. Notice the ones that are blank, like the steps that are blank here, are the ones that are intermediate steps that don't really have much control over them. Why would these steps not be the controlling steps? Like these steps over here that are blank, why are they not really controlling? I'm assuming everybody is like somewhat familiar with glycolysis. If anybody wants like a quick rundown of glycolysis, just give me a shout out. But otherwise, like the question is, why is there no like control on these series of steps?
Are you talking about? Never mind.
That's okay. Okay, so what I'm saying is like it essentially like go ahead.
Could it be because of the arrows and how it can go either direction?
Yes, that's a good thing to observe.
The feedback you're talking about, like feedback loops?
Yes, I'm talking about what am I talking about? What I'm trying to say sorry. What I'm trying to say is I'm trying to explain how you can identify the critical enzymes. And the way that you identify the critical enzymes are like this. Yes, Domelaire made a good point. F6P goes forward only. It doesn't reverse. And the reason it doesn't reverse is because I think is the correct term irreversible step? I don't recall. But the point is that the reason that these are forward and backwards is because they can just flip. They can do they can catalyze the reaction, and they can also catalyze the reverse reaction. So the reason for that is because if we need to push back and we need to do gluconeogenesis, right? We have glycolysis, which is breakdown of glycogen. We have gluconeogenesis, which is buildup of glycogen. If we need to go the other way, then these same enzymes, the ones that are blank, will do it. They will just do the reverse reaction. But these enzymes, the F6P and the G6P, are not involved because they only go one. No, sorry, the F6P is not involved because it only goes one way. Because it's a critical control point for glycolysis. So that's one way you can notice like what are the critical control points that are serving only one pathway is what I'm trying to say. So okay, what did I want to talk about? So this is a really, really nice resource because it shows you how all of it compares, how all the metabolic pathways like link to each other. So we see here that we have glycolysis. We have glycolysis starting with glucose here, and we break down the glucose all the way into pyruvate, right? Then the pyruvate gets, what is this step here showing? Does anybody remember? This step of pyruvate, why do we have two pyruvates here? We have one in blue and one in green. What happened there? Anybody know? What happens to
The PDC cycle, like when it's transforming? Is that the intermediate step that it's gonna right before it
What does the
attaches?
PDC cycle produce?
Um,
It this one, acetyl-CoA, right?
yeah, yes.
So this step, uh, wait, what is this? Why did I forget this name real quick?
I
What
for
does PYR stand for? Why did I forget this?
pyruvate something?
Pyruvate, yeah. Why did I was just say it? Oh my god, I'm skyrbrained today. Pyruvate gets turned into acetyl-CoA with the PDC. Yeah. Where is the PDC?
Outside of the blah blah.
The blah, the blah. Does anyone have an idea where the PDC is in the cell?
Well, it's not in the mitochondria, right?
By the what?
It's not in the mitochondria, right?
Isn't it in the mitochondria? No, it is in the mitochondria. What?
Oh,
Wait, is it? Wait, hold on. Let me just double check and be sure. I'm assuming that it's in the mitochondria. Dehydrogenase complex. Do. It's either in the mitochondria or on the surface of the mitochondria.
because I thought that's why acetyl-CoA has to enter. That's why it has to go through that intermediate step. But I still have to study this a little bit more. So
That's all good. It's present in the mitochondria. So yes, you're referring to something important, right? You're referring to that it needs to go into the mitochondria somehow, right? This green dotted line here is the transfer of the pyruvate into the mitochondria. You see how there's like a little border here?
yes.
That border is representing cytosol mitochondria border. Which
Okay,
side
so
is?
the intermediate step is in the mitochondria.
Yes.
Okay, okay.
Yes. So which side is cytosolic and which side is mitochondrial? The top and the bottom. We have two separate sides.
Well, glycolysis and stuff happens in the cytosol, right?
Yep.
So that's above that line.
Yes. So the top part, so here, and that's why I really like this representation, because you can easily understand where everything is happening. So we see that above, glycolysis and gluconeogenesis takes place in the cytosol. What else happens? The pentose phosphate pathway. What else happens? Fatty acid biosynthesis. What happens in the mitochondria? Beta oxidation, which is fatty acid breakdown. Ketone body metabolism, citric acid cycle.
Right.
Out of everything here, the only thing that you shouldn't really focus too much on is urea cycle. I mean, like know what that it exists and like what it does, but everything else you kind of need to know
Okay.
to a decent degree. And the starting point, like I said, is the enzymes that control the pathway, the enzymes that are driving it, like you said, like PDC complex, right? If we converted this pyruvate into acetyl-CoA, that thing is going into the Krebs cycle. It's going into the Krebs cycle now because that's its fate. It goes in the Krebs cycle, and we do all this, and we do the only thing that's not represented here well is the electron transport chain, right? So that's the only thing that is not represented too well here. But other than that, everything here is like pretty decent. Also, so if you click on the link over here, metabolic map, if you click on the link, then you can click each of these lines and each of these items and see what enzyme it is. But as you can see, the major ones are labeled here for you on the actual thing itself. So yeah, this may seem overwhelming at first, but if you just go through it like pathway at a time, it ends up being very useful because you get to see how everything links together. So that is really, so I studied metabolism by basically like making it blank and like pointing things out and being like, okay, I think this one is this, and then checking if I was right. This is this, working my way through the pathway overall until I could understand like, the fate of this might go this way or this way. It could be acted on by this enzyme or that enzyme. Once in a while, I would like try to draw it from scratch because obviously there's a difference between like recalling and recognizing. but you don't need to do that often. I think that once you have a confident understanding of like what are the major enzymes that control the pathway, that's going to be enough that like you won't always be you're not going to be asked like, what is this random enzyme here? Because it's not really significant. That's really the point. You will be asked, structures of like Krebs cycle intermediates or like anything that you see labeled here is well, not so much for like fatty acid synthesis. You will be asked like about the major enzymes in each of these pathways and like the major intermediates, but not the ones that like you won't be asked like what are all the random things in the pentose phosphate pathway or stuff like that. Things that make sense, things that you spend a lot of time studying in school anyways, are going to be things that they're also concerned about. Um, um, what else? Yeah, metabolism. Okay, so what time is it now? 6:36. Okay, good. We have a bit of time and we can do I can show you guys how I go through a biology passage. Mhm. Okay, so we went through a lot. Any questions so far? Or any questions about anything while I set this up? No, all good. One of my teachers had once said, like, if there's no questions, then you're either doing a really good job or a really bad job. So it's one of the two. Um, I don't know if we have time to go through the questions. I think I want to just get through this first part. Let's do this. Can everybody see the screen again? Okay. So this is a very, very classic, this is a very, very classic style of biochem passage. And what we're going to see here is how we actually break this down. So I want you guys to read through this first part and just I don't know, think about it, or yeah, just read through this first part. All right. So this whole paragraph here has a lot of information in it, right? When you guys see something like this, how do you approach it? I'm just curious. There's no right or wrong answers here. I'm sure you guys, if you've done some practice, you've seen like passage like this. I'm just curious right now, what do you guys do to try to understand some of these things? Specifically, I'm talking about paragraphs where they introduce like random proteins or random gene products or whatever. What is your process to try to understand it? I highlight things I really don't know. Okay.
I highlight like because it says something about homeostasis in the beginning, I kind of just highlight like where it says ratio of OPG and Rankle gene, because that balance would attain homeostasis. So I try to like just highlight the stuff that connects to the intro, even if I don't know what the gene does or whatever.
Okay.
But I automatically recognize that. So that's what I would have done.
Okay. So we're seeing some highlighting. What else? Anybody else?
Sometimes I write something on a paper, like just write something I recognize, like just a word.
Oh, just a word that you recognize to be like, okay, this is what we're talking about here. Okay.
And what paragraph it's in, like paragraph 2, because I don't try to understand like dense stuff like that right away. Because sometimes you try to do it and then there's not really a question directing you there. So I write down
yes.
paragraph 2 and then I'll write a word that directs me to it. And if I get to a question that sends me back there, then I'll go back.
That's a good. You're prioritizing your time there. Okay, that's good. Anybody else have any ideas on how we can tackle this? Like, how would we go about understanding this dense information? I take notes similar to what you did in the beginning. DHQ equals more healthy. Yes, that's also what I do. And so I like what Kenishir said about taking it, like doing it if you need to, right? Like prioritizing your time. Like if you don't need to digest all that dense information, you don't have to. But usually like they put it in there because they want to test you on it, right? So I'm going to show you how I break down like really annoying, complicated pathway stuff like this. So I break it down into relationships, but I just make a giant interlinked relationship. So what do I mean by that? Let me see. Where can I write this? Maybe I'll do it in like Paint or something. Let's see. Let's try this. Give me 1 second. Can you guys see this?
Yep.
Okay. So this is a huge paragraph, and this is like a big pain when you're trying to like if you get a question about it, thinking about it is really annoying. So I turn it into something easily digestible. So what do? First, what are we talking about? We're talking about bone resorption. So I would say, oh, it's so small. I would say bone resorption. Resorption. So that's what I'm talking about first. Bone resorption. Oh, bone resorption. So here's the first part. So the first part is talking about what it is, right? What about bone resorption? Bone resorption is triggered by parathyroid hormone under conditions of low calcium. In one sentence, I already have 3 different things I need to keep track of. So what am I going to do? Bone resorption, here's how I would write it on the test. Bone resorption, comma, parathyroid hormone, comma, low calcium. Okay, this is what I have so far. How do these three things relate to each other? How does bone resorption relate to parathyroid hormone relate to calcium?
One, so both of those pretty much keep a balance in remodeling, making and breaking down bone, I should say. The
That's true.
PTH and the calcium.
That's true.
So
Hey,
one lowers, one increase when it's too low.
which ones? Which ones lower and which ones increase?
So when it's low, I think PTH is released from whatever, I guess, the pituitary gland to
Mhm.
trigger increase, and then something like that.
Something like that, right? So let's say, so we're saying PTH was released, right? So let's say PTH increased, right? What happens to the other two? If PTH has increased, according to the first sentence, right? What would be the signs on the other two?
I mean, to keep equilibrium, it'll have to go the opposite way, right? To decrease in
What
calcium.
decreases? Decrease the calcium. Well, what do you mean by equilibrium? What I'm explaining is that according to this first sentence, right? Bone resorption is triggered by parathyroid hormone, right? If something triggers something, right? If PTH increases, it's going to do what to bone resorption? Is it triggering it? If it's triggering it, is it increasing it or is it decreasing it?
Is it decreasing it?
Bone resorption is triggered by parathyroid hormone. If I trigger a process, does the process start or does it stop?
You started.
I started, right? So we can say that there is an increase in the amount of bone resorption. Does that make sense? If the parathyroid hormone increased, then the bone resorption was triggered and increases. Does that make sense so far?
It
If
makes
not,
sense. It's just the wording that was a little tricky.
okay. Triggered by means that, or activated by means that, you know, if you think of bone resorption as the product, it's going to increase. And so if bone resorption is, so when does this happen? When bone resorption increases and parathyroid hormone increases, under what conditions for calcium? When calcium is high or when calcium is low?
Oh,
Low. So for calcium, we would write when calcium is low. Oh, I can just use a V. Okay. So what I've done right now is made a very easy to follow flowchart that helps me answer any questions about, you know, hey, what happens if calcium was high? Then I would be able to say, well, bone resorption would decrease because I've linked the items. But we're not going to stop here. We're going to convert the entire paragraph into a very easy to understand logic chain like this. Why? Because we can. PTH binds to receptors on osteoblasts, which upregulate the expression of the signaling protein rank L. Okay, so we already know that PTH is increasing, right? And PTH binds to receptors on osteoblasts, which upregulate the expression of the signaling protein rank L. So we have this item rank L now, right? What's happening to rank L? Is it increasing or is it decreasing? According to the sentence we just read, PTH binds to receptors on osteoblasts, which upregulate the expression of the signaling protein rank L. So
so
just
it increases the
yes,
expression of it.
yes. So because we just said that parathyroid hormone is increasing under low calcium conditions, that means rank L is also increasing. If rank L is increasing, what does rank L do? Binds to the receptor rank on osteoclast precursors, activating the transcription factor NFKB. So it binds to the receptor rank on osteoclast precursors. In this situation, so we see that it activates the transcription factor NFKB. So here's another thing we have, right? NFKB. And what is happening to NFKB? Is it increasing or decreasing?
It's increasing because it's activated.
Exactly. It's activated. So it's increasing, right? Then, which signals them to differentiate and activate into functioning osteoclasts? So what happens to osteoclasts? Do they increase or decrease?
Increase.
Increase. OPG produced in osteoblasts competitively blocks rank L, thereby preventing signaling. Okay, so now they've just thrown in a totally different thing, right? They're talking about something that inhibits and blocks, right? Before, I used to do a thing where I would like draw like something on top of rank L, and I would be like, okay, OPG would inhibit this. But that becomes very confusing. Let's just do it relationship wise. If rank L activity is high, then is OPG going to be low or high?
It's going to be low because its job is to block it. So obviously, if it's
Exactly. So we can say then we'll have low OPG. And what is the relationship of OPG to osteoblasts? It's produced in osteoblasts. So if OPG is low, then there would also be low amounts of osteoblasts, right? Because of this, net bone formation or resorption can be said to function of the ratio OPG to rank L. And look, we can see that as rank L increases, OPG decreases. And what does this relate to? Bone formation or resorption, right? We can see now very clearly as bone resorption increases, what happens? What's happening to the ratio as bone resorption increases? The ratio of OPG to rank L. So we have this ratio OPG to rank L, right? If bone resorption increases, first of all, what happens to rank L if bone resorption increases? Rank L increases, right? So, and what happens to OPG? OPG decreases. So we can say bone resorption leads to OPG going down and rank L going up. So we can see how the ratio would change. If the initial ratio was, 50 to 50, the new one would be like 20 to 80. We can see how we can understand how the ratio would change because we've written out the relationship. The main power of this method is that you can instantly write out every opposite method because they often will not give you, they won't give you the question the way that they have written it out, right? Let's say here they talked about increasing bone resorption, but in the question they're like, which of these would lead to decreased bone resorption? They often do this. They often are like, oh, you know all about increased bone resorption. We told you, but what if we decreased it, right? The lovely thing is you can just go back to your thing and you can just write it out by simply flipping all of the signs. You see what I did there? So they could very easily ask something like, what's the effect of what happens to NFKB if bone resorption decreases, right? And if you didn't have this method, you would have to read through all of this and manually like flip each step. You'd be like, okay, bone resorption is triggered. No, it's inhibited this time. So high calcium, PTH binds to receptors, and then you just get lost because it's hard to keep track of it. So do you see how you can not only have it, do you see how both of these statements are true? Is everybody following what I'm doing, or is it like crazy?
I'm tracking.
You're tracking? Okay. Okay, good. So this is a way for you to very easily break down extremely dense passages. extremely dense, like pathway filled passages with stuff that you don't understand or never heard of. You're not expected to know what rank L is. You're not expected to know what NFKB is. This is stuff that they put there for you to see if you can critically understand and follow along with this with novel scientific pathways and stuff, as you might see in a scientific paper or something like that. So now you're going to be able to answer questions about any of these items because you took the time to write out this. Now, here's the thing though. Obviously, this takes time, right? But it takes less time than it does struggling through the passage itself, because the passage itself is written in a way to be not to be confusing, but I don't know. They've got some wizardry about them that they know how to make a passage like very misleading. Even though there's everything here is correct, once it's time to answer the question, like there's often like a way for them to like make you feel like one thing is the answer if you didn't think about it too hard. Yeah. So this is the trick. I think I even have it on my pad that I showed you guys earlier. I think I was using this trick all the time. And I use this for everything, for like any kind of relationship thing. It's really helpful. Do I have an example here? Oh, I do, yeah. Oh, this also is actually about NFKB. NFKB, IKB. I can't even read and some kind of mutation. Yeah, it's very useful. Anyways. Oh, okay, we're almost out of time. So we might do the rest of this next week or something. Okay, so I think that's going to be it for today. Thanks everybody for showing up. I want to ask you guys, what do you want to cover in the next session? You're welcome. We're not leaving just yet. I'm just asking, like, do you guys have any questions, or do you have anything that you guys want to cover the next session or anything like that? Can we do some chemistry physics? Yeah. As much as I dread the physics, we can definitely do it.
Yeah, I also dread the physics. I just want to see how you tackle it.
I would also like to see how I tackle physics. Yeah, dude, I don't even remember what I think, like I said before, I think I tackled physics with just a ton of memorizing and like eventually like struggling hard to like understand concepts, eventually giving up and realizing like I'm never going to understand physics, and then just coming up with like absolutely metric tons of mnemonics to memorize everything. My optics flashcard is like an entire library. It's just it's not a good thing to like teach, but I mean, whatever. That's how I got through it.
I noticed that the physics section from all the practice tests, and even when I took the MCATs years ago,
Mhm.
it's basic compared to what is taught in class. Not saying it's easy, and there's like a lot of tricks that you can do even when you don't know what the heck they're talking about. And even though I hate physics, I tend to score pretty decent on my physics questions, because like I look for, like, for example, they gave me like something with capacitance when I
Mhm.
took the
Mhm.
practice test last week. And I couldn't remember the formula, but I looked at the units and I know that I had to end with a certain unit. And then so I just set up the conversion ratio and just canceled out units, and it happened to be right. So if you have like those little tricks like that, it can help even if you don't know stuff
Yeah, excellent. That's the right way to go. The units are super powerful, and knowing your units well and knowing what units equal what other units is basically giving you the equation. Like it kind of blew my mind once I realized, like, oh, I can just instead of memorizing the equation and applying it to the units, I can memorize the units and construct the equations. Um, so that's really helpful. The kinds of physics questions that I dread are the ones that are very conceptual based, especially they love to ask like conceptual based questions around Newton's laws. I hate those because they always ask like really like niche questions and I'm just like I don't understand. I don't so if it's a conceptual physics questions, it's tough. But if it's something that's calculation based, you can definitely work around it if you know your units well. And that again is just is memorization, units is memorization. So but it's not as bad as I'm making it out to sound. You can definitely do well on physics if you just have like a decent enough background. Yeah, so I think we'll do some chemistry physics next week and we'll see how we tackle it. Any other comments or questions or anything? How's everybody's studying going?
It's going.
It's going, yeah. The semester just started, so it's gonna be hard for I appreciate that everybody showed up today. It's gonna be tough, but if you can't make it, no problem. Just yeah, email me or you don't yeah or whatever, and we'll record it. I'll get the recordings and stuff set up so everybody can see the past stuff. Okay, it's 2 minutes after, so I'm not gonna keep you guys. If you gotta go, you gotta go. All right, so that's it for today.
Thank you for everything.
You're welcome. Nice to meet you.
Thank you, Ted.